Okay, so now we're going to talk about our gram-positive rods, our aerobic and facultative gram-positives. And so these are going to infrequently cause human infections in the USA, but if they do cause infections, they can cause some problems. So some are going to be associated with food poisoning, such as listeriosis, Listeria monocytogenes, some and Bacillus serraceus. Some are going to be by your spores. So we have our Bacillus anthrax and our Corynebacterium diphtheria, which both have spores. The image to the right is actually anthrax. There's also Nocardia asteroides, and I'm just going to mention this one here. This is going to be a gram-positive rod, which can stain acid fast. And this is also going to be found in the soil. It's not really going to cause a problem with people who are immune competent, but it will infect the immune compromised. Now, first up is Corynebacterium diphtheriae. Diphtheria comes from a gram-positive, what we call a pleomorphic rod, and this can form what is referred to as palisades or Chinese letter-looking growths. It's going to have depending on the species of the Corynebacterium diphtheria, it can either be aerobic, anaerobic, or facultative. Now, diphtheria itself is going to cause a grayish pseudomembrane to cover the tonsils, pharynx, and larynx. So you get a gray pseudomembrane. And this can really cause suffocation if it's not treated or if it gets too covers too much. And then also these diphtheria have exotoxins that inhibit protein synthesis. And then they have their toxin gene is going to be part of a bacteriophage. So the bacterium that can produce the toxin must be infected with a bacteriophage. Now the diagnosis is going to be based on clinical signs and symptoms. You can isolate diphtheria and then test for the toxin production. Now, of course, we have a vaccine for diphtheria, and this vaccine targets the toxin. So this is the DTaP vaccine, which stands for diphtheria, tetanus, and acellular pertussis. Now there's going to be boosters about every 10 to 12 years, and this is mostly to boost the tetanus portion of this. The antibodies against diphtheria toxin is very effective for preventing diphtheria. Now, just even though that we don't have very many diphtheria diseases in the United States, it still is going to be cause of some epidemics and endemics worldwide. And India, for example, has approximately 3,000 cases a year. So it still is important to continue immunization. You can treat an active disease with an antitoxin as well as with antibiotics such as penicillin or erythromycin. And these would be antibodies. do a combination with this guy. Now next up is Listeria. Listeria, again, is also pleomorphic, doesn't make those fancy Chinese letters that our Diphtheria did, but it is a non-spore forming, so Diphtheria can form spores. Your Listeria monocytogenes does not. They also have what we refer to as a narrow zone of beta hemolysis on blood agar, and all pathogenic strains are going to express Listeria lysine O. This guy is going to be ubiquitous in nature, meaning it's going to be in the soil, water, milk products, processed meats, feces of mammals, which also are going to include humans and birds. Now, with respect to this transmission is going to be to humans through animal contact. Contaminated food is very rare, and also transplacental. This guy can cross the placenta. So our at-risk populations are going to be pregnant women, so these are going to be about 1/3 of all cases. So they have about a 20 times increase in risk for getting a severe illness. In healthy people, they rarely develop severe illnesses with Listeria. Immune compromised, such as HIV, they have about 100 to 200 times the risk. And also the elderly are going to be at risk. So let's just talk about the disease with respect to symptoms. So you have about 1 to 6 weeks after the exposure to Listeria, you're going to get flu-like symptoms. So this is a much slower infection than would be a typical viral infection. The flu-like symptoms will be with fever, nausea, vomiting, and diarrhea. It can progress to septicemia, which has about a 50% fatality. Meningitis is about 50 to 80% fatality. It can also lead to endocarditis, premature delivery, and spontaneous abortion and stillbirth as well. The overall fatality rate of this guy is about 25%. Diagnosis is going to be culture, detection of antibodies, because again, 1 to 6 weeks, you start seeing antibodies come up. And especially if you see any kind of culture from the cerebral spinal fluid or another sterile site. Now, just really quickly about the pathogenesis. I already wrote this out so I can talk about it. So Listeria, again, is going to be able to enter your blood. It's going to get picked up just naturally by our phagocytic cells. Also, our polys, our neutrophils can pick this up. They're going to get entry into epithelial cells and hepatocytes if they're not cleared by your innate response. And then once inside the epithelial cells or hepatocytes, it's going to be able to get out of the phagolysosome because the pathogenic strains of Listeria will have the Listeria lysine O, and the Listeria lysine O is activated at low pHs. So Listeria lysine O will be activated in the phagosome lysosome. It will lyse that compartment, and it will enter the cytoplasm where it will grow. It also will be able to polarize actin, and it is referred to as an actin rocket. These will form listeriopods that will enter the adjacent cells and continue their infection. Now, treatment and prevention of listeriosis is important. So you're going to be able to treat with penicillin, ampicillin, trimethoprim with a sulfamethoazole works. It's important to cook your foods well and avoid high-risk foods, especially if you're in one of those high-risk groups. So if you're immune-compromised, pregnant, you want to avoid raw and pasteurized dairy, soft cheeses, undercooked veggies, or meats or fish. Also keep in mind Listeria can grow in your fridge, so cold temperatures do not inhibit its growth at all. There is no vaccine. And so just a little history about listeriosis is that there was an outbreak in 1981, which is the first confirmed outbreak. It was associated with coleslaw, and there were 17 deaths associated with that first outbreak. In 1985, there was an outbreak that was associated with soft cheeses, in which you had 142 cases and 46 deaths. Of those 46 deaths, 39 fetal deaths. And then in 1989, the FDA created a zero tolerance in food for regulations. And so they go around and they randomly sample plants or production facilities that make these products, such as meat processing plants, and they continue to update their regulations. There, of course, have been more outbreaks since 1985. There was a large outbreak of hot dogs in which you had 79 cases in 17 states across the United States, including 21 deaths with 2 fetal deaths as well. And that was in 1999. And there have been other, a more recent one was 2011. The Jensen farm was associated with cantaloupes. And so these people might have, these were actually fined with respect to the outbreak. So this is still a very common foodborne pathogen, and we still have to be vigilant, especially in those at-risk individuals, at-risk groups.